Hey, RGV. I'm here with Dr. Megan Kinnery, and we're going to take a quick radio break. But just remember that the insights shared during RGV Community Health Hour are intended for educational purposes and are not a substitute for professional medical advice. For personal health concerns, always consult your doctor. And we'll be right back with Dr. Megan Kinnery. All right, everyone, and we're back with Dr. Megan Kinnery. So, Dr. Kinnery, thank you for joining me today. And before we start, actually, I just want to let all the audience know that I was really surprised that, so like I mentioned before, for this month, I'm doing breast cancer awareness, trying to keep it related to that. And so, one of the first things I did was type UTRGV breast cancer researcher and then on Google, and the first name that came up was Dr. Megan Kinnery, and I was like, oh, I know her because we've been collaborating through the lab that I work in. We're doing CRISPR work. I think I probably mentioned it a handful of times here on this show on previous episodes. So yeah, I was really surprised, and I was like really thankful to know that like, hey, I've talked to her before and worked with her before and done Zoom sessions, and so I was pretty confident I could get her on. So I just want to say thank you for coming on, Dr. Kinnery, and so just feel free to talk a little bit about yourself in terms of your educational background and your journey from wherever you're from to UTRGV.
Sure. So thank you so much for inviting me today. So my educational background. So I received my BS in biochemistry and molecular biology at SUNY Albany in New York. And there I developed a keen interest in genetics
Nice.
and decided to seek a doctoral degree at the University of Oregon. So I moved out to Oregon and had a really wonderful time doing yeast genetics. And there I worked on screens. And so I worked on a synthetic lethal screen, and I did a 2 hybrid screen,
By screen, do you mean like,
a genetic screen.
oh, okay.
And so, yeah, and so it was really fun investigating synthetic genetic interactions and learning about signal transduction pathways and the cell cycle.
Mhm.
And of course, so many things that are very fundamental in biology were first delineated in budding yeast.
Mhm.
So it was a really fun system to work with. Anything you could dream up, you could pretty much do in budding yeast.
That's awesome.
Yeah, I
And
had a blast.
and so are you native? Did you grow up and were, born and raised in New York, or was that where you just went to school for your undergrad or?
So I was born and raised in upstate New York. So
Oh,
I was
wow.
born in Albany, New York. So I stayed close to home for college, and then I moved to Oregon. And I was out there for 7.5 years,
Wow.
and it was beautiful.
Oh, yes. I'm lived half my adult life in Washington State. So yeah,
Oh
the
yeah,
Pacific Northwest is like
amazing.
living in a painting. That's the
Yes,
way I always tell people.
yes, yeah. Um, and then I moved to New York City, and I worked with Ramon Parsons, and I jumped systems. And I was very interested. There was some work being done about aging in C. elegans, um, and by a Dr. Kenyon at UCSF, I believe, and I was very interested in that pathway, but at the same time, kind of in parallel, they were discovering that this pathway was also really important in cancer, and so I decided to go work on the PI3K pathway, or the phosphatidylinositol 3 kinase pathway, and what I wanted to do was do genetics, and so I had done yeast genetics, which you can do everything you can possibly think of, to human system, which is like it had its limitations, but it was just starting to really come online.
Hmm.
And so, I was really interested to do a screen, and I did, and I identified a transcription factor that modified the output of a family of transcription factors that I've focused on for over 20 years now, FOXO transcription factors.
Wow.
And it was a lot of fun, and I worked on a bunch of different projects and I learned an awful lot, and I had a great time living in New York City. And then I moved to Texas. I was a trailing spouse, so my spouse is Dr. Robert Gilkerson, and so he was hired by UTPA.
Hmm.
And he actually had several job offers, but my parents had retired to the Valley.
Oh, really?
So we were
Wow.
like, Yeah, and so, I mean, the opportunity to be near my parents was like, we were
So
like,
from New York, I'm assuming they came, they just wanted to retire down here,
yeah, yeah,
or
yeah. They loved
wow.
the weather, and my father loved to dance,
Oh,
and so he moved here to go to do a lot of dancing, and there would always be a line of old ladies that wanted to dance with my father because he was a real sweet guy. So it was really cool, and he loved it here so much, and so
wow.
he had so many friends, and so yeah, so we moved down here, and I was a lecturer in the chemistry department at UTPA for a
Wow.
year,
Huh.
which was really fun. Like, oh my goodness, though, like I hadn't taken chemistry since like 1991. Um, and so like I was up every night studying. I was like, oh
Mm,
my goodness, like,
yeah.
but it was so much fun. And then I was hired as an assistant professor in the biology department, and I started my own group. But it was really funny because like when I walked in, the Western blot developing machine was duct taped together. It would be non-functional for months at a time. It was like this Kodak developing machine. Um, and like a lot of the equipment was mostly broken. And I was like, oh man, how am I going to do research here? But I can tell you in the time I've been here, which was like I've been here, I got here in 2012,
Oh, wow.
and I've had a lab now for 11 years. The
Nice.
amount research has escalated here has been phenomenal. We have wonderful equipment now, state of the art equipment. Anything I could possibly need, I have. I have my own flow cytometer in my lab. And so pretty much whatever I want to do now, I can do. And so in the span of 11 years, we brought in all sorts of equipment. The med school came
Mm-hmm.
in. It went from UTPA to UTRGV, and this place literally transformed. It was amazing to a point where now, I have a doctoral student. I'm going to have more
Wow.
doctoral students rotating.
That's
I'm
awesome.
hiring a postdoc. And
That's
so,
great.
yeah, so I mean, really, it's been great to watch this place develop and have the opportunity to do research. And so when I was a postdoc, I discovered that FOXO transcription factors were nuclearly localized in the context of cancers where they should not have been.
Hmm.
And so, in basal breast cancer and in glioblastoma multiforme, these are both poor prognosis cancers that harbor stem signatures. And when I did my screen, I found this factor, NFIL3, that blocks FOXO from inducing apoptosis. And so we knew the FOXO factors were in the nucleus in these cells when they should not have been,
Hmm.
because the PI3K pathway is on full blast in these cells. And normally, when PI3K is on full blast, they shove FOXO factors out of the nucleus. But there's one context where that kind of, well, there are numerous contexts, but there's one context kind of at a basal state where PI3K is really high and FOXO factors are in the nucleus, and that's stem cell context. And
Hmm. Oh,
that
so it was
was,
like reverting to stem cell state in a sense?
yes.
Wow.
So when I started my lab, what I wanted to do was figure out what are the FOXO factors doing? And I had an army of undergraduates. Like I had like at least 10, and they were all looking at the known functions for FOXO factors. So they were known to mediate reactive oxygen species by upregulating things like SOD2. They were known to regulate the cell cycle. They were known to impact gluconeogenesis. And so they were testing all of the known functions for FOXO factors. And Rachel Cedillo and Krista Respondek discovered that they were regulating the stem genes. And it was like a huge signal. And so we realized that in part the FOXO factors were regulating stem genes in these cancers. And in fact, in every stem cell context examined to date, FOXO factors are driving the stem signature. Actually, this was first discovered by the Ghaffari lab at Mount Sinai, where she discovered that FOXO1 binds to and activates OCT4, which is one of the pluripotency genes.
Mm-hmm. Yeah.
Well, it turns out that in the basal breast cancer and in the glioblastoma multiforme, they do a very similar function, activating the stem signature and stem
Hmm.
genes. And
Wow.
And so then, my lab has gone on to investigate more their other roles in these cancers, and they regulate some of the fundamental developmental pathways, such as the Wnt pathway and the Notch pathway. And this work really beautifully intersects with work of other people, such as Dr. Achille working on FOXO-factors within the context of diabetes. He's also finding that FOXO-factors are regulating the Notch pathway. And so we're kind of trying to understand the mechanisms by which FOXO-factors regulate these fundamental developmental pathways and how this impacts cell fate, how it impacts the epigenetic landscape, and ultimately how this impacts human disease.
Wow, that's remarkable. So is that what you continue to work on in your lab? Yeah. So throughout
Yeah.
all these years, you've been kind of hovering around that region of investigation in these pathways and the mechanisms behind it all. Yeah.
Yep. So right now we're identifying the target genes and the binding sequences that FOXO-factors regulate, and we do it within the cancer context, where we're trying to find out what are they doing, and we love it. We've identified where they're binding. We're working on publishing that right now, and boy, we were looking at the Wnt pathway, and it was regulating a whole bunch of different Wnt genes, but there's only one that the FOXO-factors are binding, and they have a very interesting binding pattern. And so we're following up on that.
Nice.
But we also found other targets that we would have never thought of that the FOXO-factors are binding. And one thing that's very interesting, and it was something that was already known, is that the FOXO-factors are partially redundant. So it turns out they oftentimes will bind the same targets, but not necessarily at the same place. So they may have slightly different roles in regulating their target genes. And so we're working on that as well. So we work on it within the cancer context, but also in a developmental biology context. And but it's very related. Like when we initially did our gene expression profiling with the cancer cells, because I had been studying FOXO-factors for a long time, I was like, these targets look a lot like the muscle targets. Like there was a lot of overlap between them. And it's interesting because cancer has this thing called a mesenchymal phenotype, where there's this thing called the epithelial mesenchymal transition, where cancer cells become more invasive. They have these mesenchymal genes that are commonly expressed in muscle cells. And so we also study the role of FOXO-factors in myoblasts, because a lot of the targets between the cancer cells and this myoblast context are similar. And we use this myoblast context because, unlike the cancer cells that have an array of different mutations within them, and so anything that you observe, it could be a complex interaction with any of those mutations there. When you're dealing with the myoblast context, it doesn't have a ton of mutations, and so you have a much clearer kind of explanation for the phenotypes you're seeing. And so it's a nice clean context to kind of in an experimental system investigate its roles.
So it's almost like a simpler model to investigate. It's almost like doing something in yeast where it's a simple, one-celled organism versus like, a more complex, like the lab possums we use, and trying to investigate their pathways, and which would be way more complex.
Yes.
So we're going to take a quick music break and we'll be right back. Thank you. All right. And we're back. Sorry if y'all heard that feedback. That was terrible. All right. And so we're back with Dr. Kinnery. So Dr. Kinnery, do you want to talk a little bit about the therapeutics that y'all are working on in your lab in terms of how that relates to breast cancer? Or also a quick brief on, the breast cancer type that you work on and whatever else you'd like to discuss.
Yeah, so my lab focuses on basal breast cancer, and the reason being is that when I did my screen as a postdoc, this factor that blocks FOXO-transcription factors from inducing apoptosis was in a specific type of breast cancer called basal breast cancer. And it was within that context that FOXO factors were in the nucleus when they shouldn't have been, because the PI3K pathway was on full blast. And in normal cells, like say, for example, a muscle cell or a fat cell, if the PI3K pathway is on full blast, they would kick FOXO factors out of the nucleus by basically the PI3K pathway would ultimately lead to phosphorylation of FOXO factors, and they go to the cytoplasm. And so FOXO had some role in basal breast cancer, and my lab sought to figure out what that role was. And we identified a number of different roles. So they drive STEM gene expression, and we're still working on the full breadth of that project and regulating fundamental developmental pathways. And they also regulate metabolism, and we're still following up on that as well in more detail. But when we inhibited FOXO1 with a drug, AS1842856, we found that the cells quickly died by apoptosis. So they induced pro-apoptotic genes, they had caspase-3 cleavage, and they had other markers for apoptosis. And so we quickly realized that, wow, that's really weird. And we still don't fully understand the mechanism, but we have some ideas and we're following up on it. But basically, the inhibition of FOXO factors can induce apoptosis in basal breast cancer, and so we submitted patents, international, national, that are still pending.
Oh, wow. And is that part of David? Because I remember I just
Yes.
found this out with David. So David Flores, he
Yes.
is a classmate of mine in the human genetics program. And it's really funny because literally, I think it was the same day. Um, Whenever I looked you up, I was walking back to my office, and I came across David because David goes for walks around the campus. And I came across him and I asked him real quick, I was like, hey, does Dr. Kinnery really do breast cancer research? And he's like, yeah, you talked to me a little bit about it real quick. I was like, okay, I just wanted to confirm because I found when I Google searched you, I just so happened to walk out and see David. And of course, I know David's one of your mentees, I guess I can, call him since he was in your lab doing his master's.
Yes. So David was a master's student with me, and his first author publication was basically the discovery of this novel therapeutic intervention for basal breast cancer, which is
Wow.
the worst prognosis breast cancer. So breast cancer can be broken down into 5 basic subtypes. There's luminal A, which has the best prognosis, then luminal B, then there's HER2 positive, which confers a worse prognosis, and then there's the basal breast cancer, and it overlaps to a fairly good degree with triple negative breast cancer that lacks expression of the estrogen receptor, the progesterone receptor, and the HER2 receptor, so triple negative breast cancer.
Oh,
And then
wow.
there's like a normal like, and these were identified by gene expression profiling. And so basal breast cancer has the worst prognosis. And so, and so really, they don't respond to the hormone-based therapies.
Wow.
And so, but yeah, to some degree, they'll respond to like PARP inhibitors. But yeah, so inhibiting FOXO factors can induce apoptosis in the setting, and these patents are under review. We're working on the mechanisms. We know that inhibiting FOXO factors induces apoptotic genes, but we still don't completely understand the mechanism, but we're studying the mechanisms. And so,
Wow,
yeah.
it's really awesome.
Yeah, we've identified target genes. We've published target genes. We've identified where they directly bind in the genome of these cells. And so we're following up on a lot of leads to better understand, how is it that when you inhibit FOXO factors, it leads to apoptosis.
And so whenever I had my guest on, she was mentioning, because I can't remember, but she mentioned the triple negative breast cancer. I want to say that's the one she was diagnosed with,
Yeah.
but seemingly she is, she went through the chemotherapy. They weren't able to find any other cancerous cells. So of course she was going through, she might even still be there in Houston at MD Anderson going through her radiation therapy. And so, it's just kind of those last steps to try to make sure that any of those terrible cells are not hanging around. And so, yeah, it was really fascinating because I don't know anything about breast cancer. So for her to hit me with all this breast cancer information and some of what you've mentioned did sound familiar to what she was talking about. And so I just find it really fascinating to know that, like, hey, I have this awesome, collaborator that's done this type of research. And of course, my classmate, my peer knows as well. I might have to get him on the show for next. If I can't get a guest, David, if you're listening, you might be my guest next week, so be prepared. um, but yeah, and so
Yeah, so we're trying to figure out the mechanism, and it's so interesting because the genes we identified in the basal breast cancer, when we inhibited FOXO1 with AS1842856, and we did RNA-seq, we showed that it regulates the Wnt pathway, and we published that. It induces apoptotic genes, and we published that. But it does a lot of other things too that we're kind of following up on, like, for example, mesenchymal genes that overlap with like muscle cells, genes that are involved in sulfate specification, like, and like mesenchymal cells, like myoblasts, can be differentiated into things like bone, into things like muscle, and so it can be kind of driven to different lineages, just like cancer cells. And
Hmm.
so, I kind of in the back of my mind wonder if what we are figuring out in our model myoblast system, if it's going to go back and it really inform what we're learning in the cancer system too. Like, learning about how cancer metastasizes, because we're finding that FOXO factors are regulating metastasis genes when we knock them out using CRISPR. And
Nice.
so now we want to see if we put these models in like xenograft model, are they not going to be able to invade and metastasize, for example?
And so, what type of cells do you use in your lab? Is it a variety of cancer
Yeah.
cells?
So we use, it's so interesting. We use the basal breast cancer and the glioblastoma because they both have those strong stem signatures. And that's where FOXOs are in the nucleus, even though PI3K pathway is high. And that was initially discovered by Bob Weinberg, who's a famous, cancer researcher. He published that in 2008. And then through our research, in kind of an unbiased way, we stumbled upon FOXOs having an important role in these cancers, regulating the stem genes years later. But yeah, so, and in both of these settings, the FOXO factors are regulating stem genes, these developmental pathways, and we're kind of pushing forward, and where our work is really leading us is to investigate their role in metastasis and invasion.
Oh, wow.
Yeah, and sulfate specification.
And do you see any trends in the cancer or breast cancer? I don't know how closely you follow specifically breast cancer fields, but whenever my first guest came on and she was just talking about her oncologist, and like I was mentioning during the break, or maybe it was before the show, she was just like, "Oh, she just graduated medical school and she knows of all these new, therapies or technologies," and just to hear her talk about it a little bit. And actually, before you answer that, we're going to take another quick break and we'll be right back.
Okay.
All right, and we're back for our last segment with Dr. Kinnery. So, Dr. Kinnery, feel free in the next few minutes to share whatever you'd like to share in terms of the breast cancer research and trends, or any opportunities that you might know of, or things like that.
Great. So yeah, I would say remarkable strides have been made in the treatment and diagnosis of breast cancer, with all of the different, molecular capabilities of being able to subtype cancer, whether it's through gene expression profiling, genomics, so many different ways people can really better characterize breast
Hmm.
cancer and kind of, come up with a prognosis. And the treatments have really gotten more sophisticated as well. And I would say, arguably, the biggest progress has been made in the area of immunotherapies. And so there are ways that cancer cells can shut down the immunological response. And so, in our bodies, there are cells growing all the time that shouldn't be growing where they're growing in the ways that they're growing, and our immune system is able to take them out. But the most aggressive cancers figure out ways to bypass our immunological ability to take out cells that are basically overgrowing and are doing things they shouldn't be doing. And so there are these checkpoint proteins that can interact with each other and shut down T cell responses, such as like PD and PDL. And so the immunotherapies will directly bind to these proteins that are they're basically membrane spanning proteins, and they basically can take out their function. And so when you hear about immunotherapies, these are, the state-of-the-art therapies that are being given to people. But there are some drawbacks to it, such as autoimmune responses, and sometimes you won't get even if you use the immunotherapy, you might not get like what you would call like like a hot response, or getting the immune system infiltrating to the tumor site, because it could be boxed out, for example, by cancer-associated fibroblasts. And so now people are figuring out that if you target like the FGF receptor with inhibitors, you can kind of break that apart and get the immunotherapies in. And there are other checkpoint proteins too. And so it's not just PD and PDL, but there are other ones too, such as like VSER. And so I think that, but these therapies have made tremendous progress, and arguably they've made some of the best progress with treating melanoma in a fraction of patients, and in lung cancer patients. But it's also moving forward with breast cancer treatments.
And just quickly, was this a lot of this immunological work was what Dr. James Allison, I think,
Yes,
right?
yes, yes.
Yeah,
And he won the Nobel. Yes,
hmm
he pioneered that stuff.
yeah, it's really remarkable it blows me away, man, about humanity and our how clever we are in terms of like figuring these things out. Like, even just knowing about pathways, whether, being able to interact with them is a whole nother level, but just learning about the pathways and these proteins and transcription factors and these different things that interact at the cellular level, it's pretty remarkable stuff. And so,
It gives me so much hope that, I can just see the progress happening. People are identifying the roadblocks and they're going after them. And I'm very hopeful for future therapies for people.
yeah, and in part of our human genetics PhD program, this will probably stay with me for the rest of my life. And I don't remember which class it was or who was teaching, but through their PowerPoint presentation, they showed breast, like damaged from breast cancer, and then they showed the therapy that kind of helped save the tissue and regenerate some of the tissue. And it's just like, it's really like awe-inspiring to know, like, man, we can help people out from these terrible disease states. And it just takes a giant community and collaboration from everyone around the world. And that was my biggest insight into how the different disciplines can come together. When I went to the school at University of Washington, basically every department, whether it's science related or not, they just have a variety of experts in their respective fields come together, whether it's for business, the business school, or science in the science departments, and they're just trying to make great things happen. And so it's really remarkable to see that happening here at UTRGV too. So, and like you mentioned earlier, you've seen it blow up over the last few years, or over the last, 10, 12 years. And so, yeah, I'm really happy that this is happening down here because, adding all this medical research and facilities and opportunities for students is really going to change the region for the better. And so, any final words before we take our final music break, Dr. Kennery, that you'd like to share with our audience?
I guess I'd like to share a message for students. I think, stay curious. I think, follow your curiosity in life. And yeah, I think that, research can really be an engaging and rewarding thing to go after.
Yeah, for sure. And like I've mentioned before, my final note with that is don't be afraid to go to a lab and knock on the door and ask if there's an opportunity. Because when I was an undergrad, I worked in a lab, but my professor came in one morning and she said, "Hey, who wants to volunteer in my lab?" And my hand shot up as quick as I could because I thought the opportunities had to be presented to you, not that you can just go look up a professor, go to their office and be like, "Hey, do you have any opportunities in your lab or,
Yeah.
anything I can do to help?"
Yeah, my lab is has tons of undergrads. And definitely my lab is very student driven.
Awesome. Well, thank you so much for coming on, Dr. Kennery. I really appreciate your time, and I know I'll be talking to you soon about all our work. So,
Thank you for inviting me.
yeah, no problem. Thank you. And thanks to everyone. I'm going to play one last music break and then head out. But I just want y'all to know to stay curious, stay healthy, and let's thrive together, RGV.